Hello students, welcome to IURage. Today we are going to start a new topic which is Extra-Hepatic Biliary Apparatus or EHBA. So let's get started. First let's see introduction. So Extra-Hepatic Biliary Apparatus consists of 6 structures. What this apparatus consists? It consists of 6 structures. What are these 6 structures? We'll discuss it further on. What are the main function of this apparatus? What it does, it collects bile from the liver, stores it in the gallbladder and transmit it to the second part of the duodenum. So this is the main function of this total apparatus. What it does, it collects bile which is produced in the liver, it collects it from the liver, then stores it in the gallbladder and transmit to the second part of the duodenum whenever it is necessary or whenever it is needed. The bile is produced continuously by the liver, that we know that bile is producing continuously in the liver, and it is stored and concentrated in the gallbladder. So the function of gallbladder is the storing of bile, and also it is helpful to make bile concentrated. What it does, gallbladder stores and make the bile concentrated. So the bile which is producing in liver is initially diluted, and the structure of gallbladder is designed in such a way that it is not meant for secretion but for absorption. So there is no secretion from the gallbladder, it only helpful for the absorption and the storage of bile which is dilute initially. So the gallbladder epithelial cells, what it does, they actively absorbs sodium and chloride from the bile into the blood, and also there is osmotic absorption of water in the gallbladder. About 80 to 90% of water is absorbed in the gallbladder, and the concentration of the bile increases 5 to 10 times because of this. So due to this, the bile becomes thick and also dark green in color, and it becomes highly concentrated. So this is the main function of the gallbladder. It makes the bile concentrated by absorbing the sodium and chloride. What it does, absorb sodium and chloride, and also by osmotic absorption it absorbs water. About 80 to 90% of water from the bile is absorbed, making the bile concentrated, and also released intermittently when fat enters the duodenum. So whenever the fat enters the duodenum, means whenever we ingest food, the food goes from mouth through the esophagus into the stomach, and then through the pylorus into the duodenum. When the food having fat enters the duodenum, it signals the gallbladder, and then the gallbladder release the bile for the further digestion of the ingested food or ingested fat. Okay, so these are the main functions of gallbladder, which is first is absorption. It absorbs, it stores, and by absorption it makes the bile concentrated, and it releases bile only when it is necessary for digestion. Okay, so moving on. So what are the 6 structures of the apparatus? The apparatus consists of 6 structures. The first one is right hepatic duct. Next is left hepatic duct. As you can see here in this picture, this is the right hepatic duct, this is the left hepatic duct, and both these duct coming from or emerging from the liver. Okay, as you can see here, both these duct are emerging from the liver, from the right and left lobe respectively. Next is common hepatic duct. Both this duct, right and left duct, fuses together to form the common hepatic duct, as you can see here in this picture. This is the common hepatic duct. Next one is the gallbladder. So this is a structure, pear shaped structure that we have seen. This is the gallbladder. This is the gallbladder. Next one is the cystic duct. The duct which is connecting the gallbladder to the common hepatic duct is the cystic duct, as you can see here. This is the cystic duct. And the last one is the bile duct, or we can say common bile duct or CBD. So the duct which is forming by fusion of cystic duct, let me erase this. Okay, so duct which is forming by fusion of cystic duct and common hepatic duct is the common bile duct or bile duct. This is the bile duct or common bile duct. So these are the 6 structures that we are going to learn in this whole series of extrahepatic biliary apparatus. Moving on. So before starting, let's understand what is the embryological development of this apparatus. So it develops from the caudal part of the foregut. Caudal part of the foregut. So here foregut means the upper part of the primitive gut tube, which is formed during the embryonic development from the endoderm. So from the endoderm, a primitive gut tube form. Okay, from the endoderm, primitive gut tube form, and from this primitive gut tube, the upper part of the primitive gut tube. So suppose this is the gut tube. Primitive means primary gut tubes. Okay, so this is the gut tube. So from the upper part of the gut tube, the upper part is known as foregut. Upper part of the gut tube is known as foregut. Okay, so from this foregut, from the caudal part of the foregut, caudal means the distal part or the lowermost part. So let's understand it here in this picture. So this is the foregut. Okay, this whole picture is a foregut, and the from the caudal part means from the distal part. Distal means the lower part. So from the lower part of this foregut, there develops a bud, okay, which is called as hepatic bud. Firstly, what is foregut? It is the upper part of the primitive gut tube is known as foregut, which is formed during the embryonic development from the endoderm, and from the caudal part, that means lower part of this foregut, develops a bud which is known as hepatic bud, as you can see here in this picture. Let me erase this first. Okay, as you can see here, there is development of this bud from the lower part of the foregut, which is called as hepatic bud. So from this hepatic bud, again this hepatic bud divides into 2 branches, or there are 2 divisions of this bud. First one is pars cystica and second one is pars hepatica. Okay, so from pars cystica, as you can see here in this picture, this is the pars cystica. So from pars cystica, what develops? Gallbladder and cystic duct. As we have already discussed, what is gallbladder? This is gallbladder and the cystic duct. This is the cystic duct. So these two structures develop from the pars cystica, the smaller division or the smaller branch of the hepatic bud, and from pars hepatica, which is this part, this is pars hepatica. So from pars hepatica, what develops? Common hepatic duct, right and left hepatic ducts, septum transversus and hepatic parenchyma. All these structures formed from pars hepatica, which is the larger division of the hepatic bud. Okay, as you can see here in this picture, the right and left hepatic duct and common hepatic duct, these structures developed from what? Pars hepatica, from this portion, and also it helps in the formation of septum transversum. What is septum transversum? Septum transversum is mesodermal in origin, that means it is formed from the mesoderm, and also it is involved, or it is helpful for the formation of diaphragm and ventral mesentery, and it is initially present between heart and liver in the embryonic life. So you can see here in this picture, this portion is the septum transversum, and what it does, it helps for the formation of the diaphragm and ventral mesentery. Okay, so we have discussed what are the embryological development of extrahepatic biliary apparatus. Let me erase this. Okay, so firstly, the extrahepatic biliary apparatus develops from the caudal part of the foregut, Foregut is the primitive gut tube, upper part of the primitive gut tube, and caudal part of the foregut is the lower part of the foregut, and from that lower part develops a bud which is called as hepatic bud. Then this hepatic bud develops into two parts, as you can see here. The first one is pars cystica and second one is pars hepatica. From pars cystica, gallbladder and cystic duct develops, gallbladder and cystic duct develops, and from pars hepatica the remaining structures develop, which includes right and left hepatic duct, common hepatic duct, and also septum transversum and hepatic parenchyma. Okay, so moving on, now we'll start the first structure of extrahepatic biliary apparatus, which is right and left hepatic duct. So they both emerge at the porta hepatis from the right and left lobe of the liver respectively. So what is porta hepatis? Porta hepatis present at the inferior surface of the liver, and it is a depression through which the structures enters or vessels enters or leave the liver. Okay, so this is the porta hepatis, as you can see here. This portion is the porta hepatis through which structures or vessels enters or leave the liver, and it is present at the inferior surface of the liver. It is present at inferior surface of the liver. Okay, and this right and left duct, as you can see here, the right and left hepatic duct emerge from the right and left lobe of liver respectively. Okay, and the main thing you have to understand here is the content of porta hepatis. What are the contents of porta hepatis? So from it is the most common question which is asked in viva. What are the contents of porta hepatis? So posterior to anterior, if we see the content, so most posteriorly presents portal vein. As you can see here, the blue structure you are seeing, this is the portal vein. It is present most posteriorly. Then in front of the portal vein presents hepatic artery. As you can see here, hepatic artery is present just in front of the portal vein, and then hepatic ducts. The green structure, as you can see, this green structure is present, which is present most anteriorly, that is hepatic duct. So the mnemonic for this is VAD, V, A and D, means portal vein, hepatic artery and hepatic duct. These are the structure which is present postero-anteriorly, from posterior to anterior. Okay, let's understand it better. From posterior to anterior, what are the structures present? They are VAD. The mnemonic you have to keep in mind is V, A and D, which is portal vein, hepatic artery, and most in front hepatic duct. As you can see here also in the picture, the most posterior portion is the portal vein. Then in front of that presents hepatic artery. This is the hepatic artery, and most anteriorly present hepatic duct. Okay, so moving on, next structure is common hepatic duct. So common hepatic duct formed by the union of right and left hepatic ducts. So we have seen that right and left hepatic duct emerge from the right and left lobe of liver, and they emerges out at the porta hepatis, and by emerging out they both fuses with each other to form common hepatic duct. As you can see here, this is the common hepatic duct. it runs downward about 3 centimeter. Okay, so this common hepatic duct runs downward for about 3 centimeter, and it joins on its right side at an acute angle by cystic duct to form bile duct. So here you can see, or you can appreciate in the picture. So this is the common hepatic duct, which is forming by the fusion of right and left hepatic duct, which is 3 centimeter long, and at its right side by an acute angle, it is fusing or joining the cystic duct. So this is the cystic duct, and this common hepatic duct joining the cystic duct at an acute angle, and both this duct, which is common hepatic duct and cystic duct, together makes the bile duct. Okay, so what is common hepatic duct? It is the fusion of right and left hepatic duct. The fusion of right and left hepatic duct. The common hepatic duct runs downward for about 3 centimeter, and at right side by an acute angle it fuses with the cystic duct, and both this duct forms bile duct or common bile duct. So again, one important point you have to keep in mind in this lecture is there are also accessory hepatic duct which are present in liver, accessory hepatic duct, but these hepatic duct are present only in 15% of subjects. Why only 15% of subjects? Because of the embryological variation during early stages of development. So what exactly happens? As the liver, gallbladder, wait. So as the liver and gallbladder and common bile duct, as these three structures develops or grow during early stages of development, there are small ductal channels formed during growing of these three structures, which is liver, gallbladder and common bile duct. Small ductal channels formed. Okay, the small ductal channels like this are formed, and over time they regress or fuse into it. These ductal channels, what it does, they regress or they fuse into each other, and or right and left hepatic duct, they fuse in either into the right or left hepatic duct. But failure of this fusion, failure of these small ductal channels will lead to the condition of accessory hepatic ducts, will form these accessory hepatic ducts. As you can see here, this portion, these are the accessory hepatic duct, you can see here. So when the small ductal channels fail to fuse or fail to their fusion, will lead to the formation of accessory hepatic duct in the liver, which is a normal condition, but it has an surgical importance. Although it is present only in 15% of subject, it is still a normal condition, but the main complication come during the surgery, because during cholecystectomy, that means the removal of gallbladder, the failure to identify these ducts, these accessory duct, or to misunderstood it with cystic duct, will lead to the accidental injury, okay, or ligation. Because if there is accidental injury during surgery, this will lead to the bile leakage from the liver directly outside the liver. If there is any accidental injury, let's understand it properly. Okay, so if there is any accidental injury in this accessory hepatic duct, the bile which is producing in the liver will go through this and directly leak into the outer side of the liver. And if there is ligation, accidental ligation of this accessory hepatic duct, it will lead to the biliary obstruction. So for this, pre-operative imaging is necessary. Okay, as this accessory hepatic duct starts from the right lobe of the liver and they also terminate into the gallbladder or common hepatic duct or also upper part of the bile duct. Okay, so these accessory hepatic duct terminate into gallbladder, common hepatic duct and upper part of the bile duct. And also it is a normal condition, but only complication comes during surgery. That is, accidental injury can happen, and due to the accidental injury the bile leakage happens. And also if there is accidental ligation by misunderstanding it with a cystic duct, there can be accidental ligation of this accessory duct, which will lead to the biliary obstructions or segmental biliary obstruction. So I hope you understand what exactly is extrahepatic duct. So that's all for today's lecture. In the next lecture, we'll study the remaining part of the extrahepatic biliary apparatus. Until then, keep reading and don't forget to subscribe, like and share. Thank you for watching this video.